I will talk about, as I said, functional imaging and anatomy, but also show you a transition which is currently happening with using high field imaging from more like areas and network resolution down to what's happening also inside brain areas. So I will show you this transition that I will, in the first part of my talk, relate a bit very similar methods as we heard in the last two talks to identify probabilistic maps of areas and networks. Then in the second part of my talk, I will go into more detail and talk about how can we look, what happens and what is the anatomy, also the functional anatomy within brain areas. Because it's important to know where are brain areas, but then in functional imaging, with high resolution imaging, we want to also try to understand representations inside brain areas. And this can be topographic representations, like in retinotropy we heard in the first talk, but it can also be at the level of layers and columns, as we heard in the second talk, but all defined more indirectly using functional probes of features and representations. Then I will detail what we can do nowadays at that high-resolution imaging level, where we go in sub-millimeter functional data resolution. And then finally, I will talk about also how we relate it to columnar level, but also laminar level imaging. So the first point I want to stress is that a big goal is, and that links my talk a lot to the previous talks, that we try to relate what happens at the macroscopic scale, and in the functional space we want to go up to the level to understand the conscious mind. So how is the mind working? What is the neural correlate of the mind in the brain? And of course for that we need to probe the functional regions of the brain and what they are doing to support the mind, and relate then and establish a mind-brain relationship. But of course, this will be very superficial if we only stick to very coarse regions. I cannot understand the thought or kind of content of my mind if I just look at very broad parcellations of the brain. To go closer to content of my mind, I need to zoom in, and therefore we need to go to more fine-grained levels. And I will focus today first at the areas and networks, and then later on I zoom in and go at that level where we can relate within a brain area regions across layers and columns to content states of the mind. And of course, ideally we want to go further down up to the nanospace, but my talk will cover actually these two talks, and the later talk, especially from Carl Zillis, will of course focus also more in the much more high resolution scales. So these two scales will be now a little bit covered in my talk. This thing doesn't work well, just to tell you. Right? So if I press 10, okay. So here we see a very cross layout of areas which are described by functional brain imaging, which relate to anatomy, of course. And the difference between the previous talk is that instead of having cytoarchitectonics to identify in different brains the same corresponding region and then look how they are across brains located in probabilistic way, we probe the brain by functional tasks. So we kind of stimulate the brain or ask the subjects to engage in certain mental tasks, and then we look at the activity in these brain 1, brain 2, brain 3, and try to relate this again in a probabilistic atlas. That's the first part I want to show you. And one point I want to stress is also that we have different possibilities how to align brains, right? Because one is, of course, we bring the brain in a common volumetric space, like MNI space or Tellerac space, and then average across corresponding coordinates. But I will also use in my talk mainly a little bit more interesting methods which go beyond a simple volumetric standard space alignment, but focus on cortical alignment. So just to show you why that is important, we heard very nicely in the previous talks that there is not a perfect relationship between function in the sense of cytoarchitectonic areas and sulcal-gyral boundaries, but there is some. In the calcarine we saw the upper-lower visual field is nicely related to the middle of the calcarine. In Broca, we saw in Katrin Ammund's talk, it's not so perfect, that the sulcal boundary and the areal boundaries. And we have even a more indirect way, because we have not the real site architectonics, but we have functionally probed areas. So it's another level of variability. But we can study that. That's the point. We can investigate how variable are these kind of functionally mapped areas across subjects if we use either a standard volumetric alignment or using a little bit more sophisticated cortical alignment. And the cortical alignment has the advantage to more explicitly bring corresponding sulci and gyri in a real matched space. For example, here we see that if we use a standard volumetric alignment, here in Talairac space in this example, then we see that the same coordinate, this red cross here in two brains, these are two different brains, is one in the anterior bank of the central sulcus, which you see here, and in another brain, it is in the posterior bank, you see here. So kind of, although it's the same coordinate, it references different regions at the macroanatomical level in the two brains. And of course, a cortical alignment is not perfect with respect to site architectonics or to functionally probed areas, but at least it might remove some of these variabilities that you, with the same coordinate, mislocate across subjects locations if you use these kind of rather simple alignments. So the idea is to use, oops, that was my mistake. The idea is to use cortical alignment, and I just show this highlighted here by lines of sulci of different subjects. So different colors are different subjects identified, central sulci and many others. And if we plot them in a volumetric space, extract the sulci and plot them on top of each other, then we see how variable the locations of these sulci are. If we run a cortical alignment, which kind of co-registered corresponding sulci and gyri with each other, we get a much more nicely corresponding thinner region per sulci, which means all the subjects cluster now together. That means we have established a much better correspondence between matching landmarks across brains. That's the idea of cortex-based alignment. And here's an example how this then technically works. I will not go into details, but the idea is that the cortical folding, the curvature calculations, is brought on spherical representation. And you see that this curvature information, like this is the central sulcus, is in the superior frontal and so on. You see that although it's now curvature which is shown here, you see the macroanatomic information, because the curvature is quite similar across brains, not perfectly the same. Therefore, we slightly smooth also the curvature. But in principle, we see then how this looks like. And it looks a bit washy here, because this is bringing a kind of standard MNI or Talairac aligned brains on the same sphere. So several ones is a probabilistic curvature map. And if we then do the cortical alignment, it means we kind of move the sphere such that the corresponding curvature is maximized, and then we go back in 3D, then we see that it looks now almost like focused, unfocused focused. And that's the benefit of cortex-based alignment. You see this from this to this, or for the other hemisphere from here to here, right and the left hemisphere. So therefore, that shows that we can then identify landmarks much nicer, and we can project the resulting alignment back into 3D and have each brain, sulci and gyri, also three-dimensionally corresponding with each other. That's actually done here. This is one of the early examples in our lab where we did that. And we now assessed what is the benefit of doing CBA as compared to standardized volumetric alignment to Talairac and MNI space. And what we found was very interesting, and actually alludes a lot to what we heard in the previous two talks, namely that some areas, like for example the frontal eye field, after cortical alignment, we have a gain of overlap across subjects as compared to volumetric alignment of 100%. So we are much better. You see this in the white color. It means all the subjects kind of, after aligning the connection of the precentral sulcus and the superior frontal sulcus, we get almost perfect correspondence here across subjects that we have the same area at the same macroanatomical location. This is functionally probed frontal eye field by an eye movement task, right? And if we do this for other regions, like we heard from Katrin Ammund's in the Broca's region, there's much less gain. So that means not only the site architectonics, but also the functionally mapped speech areas do not perfectly benefit from aligning the macroanatomy. So there is a high variability how good or how much macroanatomy helps to align function. But you see, this is all positive. Positive means there's always a gain. In each area we looked at, there is a gain as compared to volumetric alignment. So that speaks that it's better to use cortical alignment also for improving functional alignment, although it's not the same quality in all parts of the brain. That's what I want to show with this slide. Here's a work, actually, this is also in collaboration with Katrin Ammunds and especially Mona Rosenke, who's actually here in the audience, and is a work mainly driven by Jülich and also Stanford, by Cullen and Krill Spector's lab. And what was shown here was to use exactly this cortical alignment for the cytotonic map. So the same idea I just described, but applying it now by first doing a macroanatomical alignment, or here, for example, not only the whole brain, but here you see it around the calcarine. So imagine the calcarine sulci are perfectly aligned in the sense of cortical aligned. Then the probability maps are actually looking almost perfect across subjects, because V1 cytotonic areas, the early ones, respect quite nicely the macroanatomy, and therefore you get a very, very good probabilistic map of the early visual areas. Here the cytotonic areas, and here the functional areas, which match actually pretty well. So the occipital 1 here matches well with V1 and so on. So that's actually, again, showing that using cortical alignment helps or benefits an alignment of cytotonics. And we apply the same idea to functionally probed areas. So here's a new functional atlas coming in a moment, but this is the anatomy behind. So this is kind of from 19 brains, again, work done mainly by Mona Rosenke, where 19 brains are cortically aligned and then averaged, and you see this is how then the average brain looks like. So it's not a single subject brain. The quality is quite high, but it's 19 brains in one brain, keeping, because the macroanatomy is aligned, keeping the quality almost like a single subject brain. Of course, it's a little bit smooth, but it's remarkable how good you can see all the landmarks which Svenja Kaspar so nicely showed in the first talk. And then, of course, the important point is we now wanted to build here in this collaborative work a probabilistic map of visual areas in the occipital and temporal lobe, and especially also at the ventral surface and of the brain here. And you see now, of course, that across these 19 subjects, we get the early visual areas exactly looking like in this probabilistic map, like the study done with the cytotonic areas. But we also get many more regions. For example, place regions, face regions. Face regions are colored red. Body-related regions are colored green. MT sits here, and so on. So we get a very nice parcellation now of this probabilistic map using the same mathematical ideas like in the cytotonic probabilistic maps, just that the areas are probed by functional tasks. So we stimulate by showing people pictures of faces, houses, places, body parts, and so on. And here's this on the ventral side. You see here the face regions, place and body regions aligned. And these surfaces will be also freely available soon for download, so that we can also add this and hopefully integrate also in the human brain atlas. We also not only use this, it is the software I have developed, Brain Voyager, but of course we put this also in more widely used software like FreeServer. So the same information will also be available in the FS average of FreeServer. So that's fulfilling this kind of standards. Then I want to move a step further and look inside brain areas. So if we know where V1, V2, V3 is, as we have also heard in the first talk of Svenja Kaspars, we have also topographic information inside brain areas. For example, in V1, we have retinotopy, as was nicely described before. And now I want to show you, if we would know the location inside V1, where the visual field is mapped to which location, we can use this for very interesting functional experiments and so-called decoding experiments. So using that knowledge by using now functionally probed retinotopy, where we identify functionally the early visual areas, but also look how topographic information spreads inside these areas, like from foveal to periphery, for example, then we can use this very nicely to decode information. For example, in our case, we want to decode what's in the mind's eye of a person. So if a person imagines seeing a shape in front of himself, just mentally, it's not really shown, can we show to the person what he has in his mind, right, as a topographic image? And for that, we do the so-called PRF, population receptor field mapping, which is just a more model-driven way to identify the retinotopy of early visual areas. But then we inverse these tasks and look when certain anatomical parts become activated by pure imagery, by just imagining seeing something, we decode back from the known anatomical region in this map back into the visual field. So we highlight the regions which are active here. This automatically actually encodes and decodes the cortical magnification we have seen, because we train the network in kind of Cartesian visual space, and we get then a decoding automatically again in that space. So we have not to do anything. The images will just look like real images afterwards without distortions. And that is really based on anatomy, because we first have to exactly know how we map the external visual field on the retitopic cortical topography. And then if here's something active, we can reconstruct the image and even show it to the person in the scanner. We actually built a brain-computer interface for locked-in patients where we want that they can just imagine letters of the alphabet one after the other, and we can decode this to build a BCI system. This is a European-funded grant. If you look at this, how this works, actually works already pretty nice, although these are only 4 letters we tested in the first preliminary experiment. So subjects here see these letter shapes. First, it's not imagined, it's just seen. And then from the activity in the visual cortex, we get activity and we decode it back in the visual field. So these are reconstructed letters, which from primary visual cortex, where the information doesn't know anything about this letter, but this comes then out, and you can almost read that like an HTSC. So that is possible now using 7 Tesla MRI to decode that. You can even do this in 3 Tesla, but this you cannot do. We tried. So we need high field imaging and high resolution and high signal to noise to do that, at least for single trials. So if a subject now not sees this letter, but imagines seeing the letter, there's nothing shown, then we get something which looks not like nicely like this, but it's more like an H. If someone imagines seeing a T, we get something which looks more like a T than at least this one. And I have to admit that the S and C are a little bit tricky. So the imagined curved shapes are not so clear in our subjects. But we use some fancy methods like deep learning methods, autoencoders, to let the networks, so-called deep learning networks, learn from these representations. So when people see these letters, we tell the deep learning network from the brain activity that corresponds to representations which correspond to this one. And then we let, when people imagine that, let such an autoencoder reconstruct or denoise the representations during the mental imagery, so they get more similar to the ones of the seen ones before. And if we do that, we get also from the imagery readable letter shapes, also especially from the curved ones. So that's now what we can reconstruct when people imagine letter shapes, and they look really readable. So this is reading, so to say, the image in the mind's eye of a person. And of course, we will now in the future go further and do this for all 26 letters of the alphabet and test it really on locked-in patients as a new form of BCI. Now I come to the second part of my talk. And what I said now was mainly, except the last experiment, but mainly related to conventional resolution imaging. On 3 Tesla, people use typically something like 2 millimeter, a bit more, a bit less. That's the resolution used there. But 2 millimeter is not high enough to resolve cortical layers functionally and cortical columns. And for that, we push, in our lab, but also many other labs at the moment, we push the limit of functional resolution down to sub-millimeter space. And that little bit, that looks not much, but it's quite difficult to achieve with fMRI. But the benefits are huge, because we suddenly not only see areas and networks, but we see also structures inside the depths of the cortex, and also tangentially along the cortex. We see within the same area feature codes, like representations which have representational information. Just to tell you, if you do that, you get very tiny voxels. When we zoom in this tiny region here around MT, you see that the voxels, these are original data, basically except a little bit of denoising of drifts in the data, raw data, not even spatially smoothed at all or motion corrected, so really raw data. But you see basically you get now 2 different colors and very tiny voxels, and these 2 different colors, we will see later, are akin to different functional responses inside this brain region. So unfortunately, we cannot do this high resolution with 0.8 millimeter functional resolution in the whole brain. We usually use special sequences like grays, which is a mix of gradient echo and spin echo, to be not so driven by the draining veins, which kind of a little bit deteriorate the quality of fMRI. And other techniques to clean the data, to not interpret data close to vessels and so on. So it's quite some sophisticated work to make the data good enough. But if you do, you get, again, benefits like seeing structure you cannot see on a 3 Tesla 2 millimeter resolution. For example, here, this is actually not yet cortical layers, but it's again topography, but now not at the huge V1 cortex, but in a tiny nucleus, the inferior colliculus. Actually, this is from an auditory probed experiment, the same as Svenja Kaspers has shown before in tonotopy. Here it's probed the same kind of different sounds, frequencies of sounds, but mapped on an inferior colliculus, a very tiny structure. Where in 3 Tesla you get 1 or 2 voxels, which show you that you have some activity there. But now with this high resolution, sub-millimeter resolution, you see a whole tonotopic map inside this tiny region, right? So therefore, that is a first big benefit you get. You see topographic information, which you cannot see with low resolution functional MRI. The next thing is, and that's the goal which drives our lab forward, we want to understand the representation of features in the cortex. For example, what we actually want to achieve is something a little bit less high quality, of course, but something like invasive optical imaging. That is the goal we want to develop fMRI into. And why is optical imaging so good? Because you see inside areas feature representations. For example, you see not only the retinotopy with that high resolution, here this is from optical imaging, you see basically these kind of patterns here, which are different color codes for mapped feature representations of orientation, which is coded in early visual cortex, here in V1. So you see that we get beautiful maps with this so-called pinwheel structures around which we get all orientations and forming a so-called hypercolumn, which is then responsible in one hypercolumn for all the orientation features of one particular position in visual space, a tiny area in visual space. And the next kind of hypercolumn with all orientations and the next, they all cover what we see in retinotopy, but we have now the high resolution to see inside each point of a retinotopic map, what are the features inside? And there are actually some more. These are just some of the more prominent ones, which go back to the Nobel Prize awarded work from Goebel and Wiesel, which found these kind of orientation columns, which penetrate in depths you don't see here. You see here would be the depths, but this is the tangential view on the cortex, and you see these kind of nice coding principles there in optical imaging as a map. And we want to do this with 7 and even higher field of MRI, of fMRI. And the idea is that we exploit the fact that we reach with 7 Tesla a resolution which is more at the level of sub-millimeter as opposed to 2 millimeter and 3 Tesla, having still enough signal to noise to map these features. But of course the problem is, or say it the other way around, we are very lucky that the resolution of these structures are not at the resolution of neurons, because then, of course, fMRI would never be able to map these features like orientation features, vertical features, and others. We are lucky that the brain is organized, again, at a certain spatial scale in this cortical columnar fashion. And that's a luck for us, because the width of these columns is depends on where we are in the brain and which type of column we are looking at. But the width in visual cortex is between 0.5 and 1.5, depends what we do. And that means they are approachable from the small voxels of high field functional imaging. I want not to say that the resolution is good enough that each voxel is precisely one column. That's not at that level. But the voxels are so small that they are more and more driven by individual features, which might look like columns or not. But the functional separability at the feature level is coming up in these high resolution images. And if we exploit this now, if we go into this mapping, we have had here actually the first convincing study for orientation columns. So this looks like these pinwheels, almost like we saw in the monkey invasive optical imaging. But here this is human brain imaging and using 7 Tesla spin echo sequence, where we see now the orientation columns mapped in the primary visual cortex of a human brain using 7 Tesla MRI. The problem with that approach here was that with the spin echo and the modeling, they could only analyze a slice which was very straight along the calcarine. Actually, we see this here. So they had just one slice in that study, so that they had a quite big resolution in depths of 2 millimeter, which is not bad because in depths you anyway have in V1 the same feature vertically, so you just average in the same column in depths. But along the slice here, which you see here, the resolution could be pushed to 500 micron, which is amazingly high for functional MRI. So they could then see the columnar structure. But the problem is this technique cannot be used in curved folded cortex. So the researchers selected this kind of straight calcarine data here. So what we wanted to do in our lab is to develop techniques where we could analyze any curved cortical patches with the same kind of analysis. Actually, we added the resolution in both directions, so tangentially without averaging and vertically without averaging, so that we can now interpret the data in both laminar resolution as well as in columnar resolution. Just want to point out that, of course, in the real data, you find not these textbooks columns, but you find much more little bit messy structures. But that is okay, because we can model that very nicely, as I show you in a moment. The first important point for modeling is we can no longer assume that the cortex is a flat sheet. If we inflate it like in the cortical alignment, we had just assumed the cortex was one surface. But of course, if we go into this sub-millimeter resolution, we have to realize that the cortex has a thickness, and then we model in the two banks this with multiple surfaces or more involved mathematical structures to basically be able to model that. I will, because of time, skip that slide, but here I want to point out that another complexity comes from anatomy, because originally in our first papers, like 5-6 years ago, we really were a little bit naive to anatomy and assumed that the layering is constant across the cortex. So layer 4 is always at the same relative position in depths, which is, of course, not true. It varies across areas, like we have seen in Katrin Ammund's talk, but it's also varying even within the same area, depending on the curvature of that brain. So if your area is a slightly different location, where there's less or more curvature, the positions of the layers shift. For example, if you have here this nice fundus of a sulcus here, then the layers here, close to white matter, inferior layers, they will be pushed and squeezed and become thinner and move downwards, so to say, whereas here in the crowns of gyri, the layers move upward. So it's a little bit like the Fliehkräfte, like the side forces, if you go by a car, which pushes you to one side. And so it turned out that the best way to accommodate this is to use a so-called equivolume principle, so that even despite curvature, you want to keep for each layer the same volumetric space in such a corresponding field. For example, here you have a longer elongation along the pathway of the gyrus here, and then you can make the layers thinner, because then you get the same volume, whereas here you have just in that direction a tiny pathway, but then you make the layers thicker, because then you get the equivolume like here. So these volumes must match, right? So if we use that principle, we basically can then sample. If we measure and we know the curvature in the cortex, we can then measure and select the laminae at the right depths, because we do it curvature dependent. You should know we don't see these layers in normal fMRI, right? So we have to use mathematical tricks to be as close as possible. Actually, again, I will not go into details, but we model that in our tools. So we use this equivolume principle, and we also validated that on a 9.4 Tesla study, where we had a resolution of only 350 micron, three-dimensional resolution, and we could see very nicely in the raw images the Genari stripe, which we have heard before as layer 4 you see this actually nicely here. This is layer 4 in a primary visual cortex, and actually also here. And if we see that, we can measure the relative position and depths of that cortex, and we can use different mathematical tools to estimate then layer 4. And if we do that, we see that only if we use the volume approach, the new approach, the kind of straight lines here, we get an estimate always at the same position, independent of curvature, where the Genari stripe is. So if you go through V1 at different curvatures, concave, convex, straight, these are the different curves here, 3 curves, then we can estimate always at the same location the Genari stripe, and that means layer 4, so it's constant. But of course, in reality, if we would not use equivolume, this is the stippled line, we see that indeed the layer 4 moves downward or upward depending on curvature. right? So here straight is quite at the same position if the curvature is straight, but if we have concave and convex curvature, it's shifted in the expected way. So it's not only a theory, going back to Bock's principle, very early anatomist, but it also shows as real information. So if we use all these kind of samplings, we can then put and make these kind of columnar maps of the cortex. And I give you just two examples, one of our lab, one of a lab from Tutel, from Boston. And if you do that, you see here, we have mapped, this is 20 years ago, area MT, which I will focus now, V5 in the human, by motion stimuli. So just showing at that time, in the 90s, the early days of fMRI, you were happy if you see something moving versus static on 1.5 Tesla actually, not 3T, this was 1.5 Tesla. And you see V5 coming up mainly if the dots move and does not come up when the dots are static. So it's a nice, simple localizer of motion. But at that time, no one asked for columns. At that time, we were happy to get this contrast, right? Now we ask the question, can we map the features, which would be direction of motion, using now high-field imaging with the techniques I have just described? And I wouldn't, of course, say you that if it wouldn't be possible. So we got maps like 5 years ago, the first extrastriate columnar maps in the human brain in MT, where the colors now show you not orientation like in V1, but the colors show you direction of motion, so-called axis of motion going in two opposing directions. And these colors basically give us the first maps at different depths maps here, showing that they are not perfectly, but pretty consistent also across depths. So these are columnar maps in an extrastriate area MT. Even nicer is that if we select the mapped features and look at their time course, how they look, cross-validated means you fit this in some runs and then look how these look like in other runs. You do cross-validation. You see that indeed we get beautiful tuning curves with a unimodal peak at the preferred direction of motion, and other directions of motion show much less activity, which looks very similar as what has been described in the macaque monkey in MT. Now I want to show you a work, not in our lab, but in the lab of Roger Tutel, which very nicely mapped two other features in extrastriate cortex, in V2 and V3, namely the so-called thin and thick stripes, which are actually found invasively by oxidase staining. You get this sort of chrome oxidase staining, and if you find then thin and thick stripes, which basically correspond to color-selective parts of V2, V3, and thick stripes for binocular disparity. But this was not shown yet in functional imaging in the human. By using, again, 7 Tesla high-field imaging with a resolution in that case of 1 millimeter, they could find these kind of stripe patterns. Here's V1, and this is beyond V1. And they look very nicely striped as expected from this set of oxidase staining. And if we look in more detail, we see indeed that here's V2 and V3 on a flattened representation, and we see here the more thin stripes, so to say, for the color representation, and the more thicker structures in between for the binocular disparity, two features which are also coded in visual cortex. And you see they're really interdigitated when we put the thick stripes, I mean now it's functional data, of course, disparity data, next to the thin stripes, so to say, so the color mapped regions, we get a pattern which looks quite similar as has been described in the macaque monkey. These are just examples that this is really possible nowadays to map features. But I should also be careful by saying one thing about limitations. At the moment, all which has been done was mapping and showing that what we know from the animal, from the macaque monkey mainly, can be found in early visual cortex, mid-level cortex. We have just published a paper on STS for multimodal integration. So that works pretty well. But the point is, the real challenge is to unravel feature codes which are not known. So if we go, for example, in the face area, which actually we work on right now, we want to understand what are the coding principles of features which build up faces. Is it holistic? Are the feature parts represented there of a face? Is there spatial relation information? And then we are no longer can go to the monkey and have the maps already done for this. We have to really develop new maps, and that's the challenge for the future. But now I want to show that we can do some amazing things in my last slides. Just show you that we can already do cognitive research by exploiting this high resolution of columnar structures, but also of laminae. I'll show you an example for both. So the first is if we understand these cortical columnar representations across the tangential extent of the cortex, we can basically now try to see how these change when the content of the mind changes. So that would relate content of the mind to different activities within brain areas of the features represented in these areas. This I want to show you now in one example. This is an ambiguous stimulus. Actually, there are just squares shown either here, and correspondingly here and here. And you might perceive a horizontal motion, but you might also perceive a vertical motion. Because when you look at this for some time, you might see a switch of vertical versus horizontal motion. And now the interesting point comes, the whole motion you see is not there. Your brain reconstructs that. It's a constructive process of your brain. Your brain tries to make sense out of that stimulus, and the best sense of these apparent motion, that kind of the dots jump are here and here, is to fill in a motion there. But since the distance is made in a certain way, you either your brain fills the motion like horizontally, or your brain fills the motion vertically. And that switches back and forth between 5 and 10 seconds, which is perfect for fMRI because we have a low temporal resolution. So the switches are not like a Necker cube very fast, but quite slow. And that we can then look in the brain whether these different percepts, which are very private individual, you all have to switch at different moments in time, whether we can relate that to the cortical columns in MT, which I just showed you before. Can you turn this on? It's jumping out of the presentation. Just play. Maybe not, because that. Yeah, thank you very much. So if we now look what's happening, then we see that we map this also physically to know by real moving horizontal and vertical. So we knew the columns for horizontal and vertical. And then we looked what happens when subjects report by button press. I perceive this ambiguous stimulus now as horizontal motion or now as vertical motion. It's the same physical stimulus. Then we see that the columns activity changes. So when the subject says, I'm seeing now a horizontal motion, then the columns for horizontal went up in activity and for vertical down, and vice versa. So you see that the switch of your percept can be explained from the dynamic activity changes in area MT. So you can relate the content of your mind to activity patterns which dynamically change within a brain area. And that shows that we can now reach that kind of resolution. And this is really columnar. Actually, the physically mapped columns and the ones active during during ambiguous motion actually are corresponding quite nicely, and they have really a nice columnar structure. The green is for horizontal and red for vertical. The other way around, sorry. And now, just 2 more slides. The last example I want to show you is that we not only can analyze the columnar structure, but we can also analyze laminar structure, laminar patterns. For example, here's an experiment done by Lars Muckli's group in Glasgow, where images were shown to subjects, and from the activity again in V1, was decoded using multivoxel pattern analysis what the subjects have seen. For example, these different images here. And when the parts of the visual scene was occluded, the subject fixates here, so it's one quadrant occluded, then they could still decode what the subjects see from the activity in the visual cortex where no stimulus was. How is this possible? So you use a quadrant which is not stimulated, it's occluded, right? But you can still decode what the subject sees, what scene the subject sees. And the idea is that the information from the surrounding goes to higher visual areas, is completed there to a full scene image, projected downwards, and the top-down activity also stimulates this part of the visual cortex which is not stimulated. So it's a top-down effect. And we reasoned then that this top-down effect should reach the upper layers of the cortex, not the middle and lower, because the middle layers are for input processing, not for top-down processing. So we mapped, using 7 Tesla again, we mapped here in the visual cortex the depths of the cortex again, and then looked and had classifiers, decoders, support vector machines, which basically get not the whole activity, but just from certain layers, like the lower layers, the middle layers, the upper layers. And we found that only the activity from the upper layers. So this is when the full image is shown. These are the cortical depths, middle layers, upper layers, lower layers. And you see that if the full image is shown, all layers can classify middle layers, upper layers, what a person sees. But if we use this image here, where this is occluded, It's the middle layers have chance level performance, and only the upper layers activity data can classify almost as good as if you see that information here. From that activity here can classify what a person has seen. So that speaks to the point that the top-down information indeed stimulates or comes down to the upper layers of the cortex. So we can now also use fMRI at that resolution to understand the input and output into brain areas by separating cortical layers. With that, I want to finish up and just summarize my presentation. So you have seen that in the first part that we need to relate different levels of the brain, and that also this macroanatomical cortical alignment seems to be useful because it improves the correspondence between subjects, whether these are cytoctonic areas or whether these are functionally probed areas. So it's an important tool to use for group studies when we parcellate the brain. I have also shown to you that with ultra-high fields, 7 Tesla and beyond, we can bridge the macro and the mesoscopic scales because we can look inside identified areas at the macroscopic scale and then can look into the columnar and laminar patterns of activity using high field imaging of the same regions. And we also have shown that we can map known features also in the human brain akin to what is done invasively in the macaque monkey, for example. But of course, we have a challenge ahead to find out what the columnar code or the features are in areas which we don't know yet the features used by the brain. And the challenge is now to identify these features. And we have shown that this is possible, but it's also possible to separate laminar profiles, which is important to understand better cognitive processes like attention, top-down activity, completion, and also connectivity, functional and effective connectivity in the brain. Because if we separate middle and upper and lower layers using brain activity, we can relate what's going on better to effective connectivity, because we know from the laminar profile whether a certain area in a network is driven more bottom-up or more top-down. And this information is difficult to get using purely functional tools like Granger causality or even dynamic causal modeling, because you always rely on assumptions. If we can use laminar profile to know this is a middle layer and it's now input activity, or an upper layer, this is more top-down activity, we can make the arrows on our models much easier by more profound knowledge, which goes back to anatomy. Thank you very much.
Yeah, thank you very much, Rainer, for this macro to micro relation.
Thank you.
Are there any questions right now? Carl?
This top-down activity in the superior